Yes? So we are now moving into the second half of the genetics unit. The first half focused on bacteria and biotechnology and kind of the genetics of bacteria and how we have taken advantage of some genetic mechanisms within bacteria to help us develop some of those different technologies. The second half of the unit is going to bring us back into what we more traditionally think of as genetics. We're going to get into some Punnett squares and dominant and recessive and all that good stuff. That is kind of fun if you're one of those people that thinks Punnett squares are fun. And we're going to talk about some different types of inheritance. So that's kind of what we're going to be working on through the second half of the unit. To start with, we're going to watch just a short, it's like a 4-minute video that will hopefully refresh your memory on some of the basics. As you watch this video, you should have a basic idea of who Gregor Mendel is and remember a little bit about why he's important. Also listen for the basic genetics vocabulary, words like dominant and recessive, homozygous and heterozygous, genotype and phenotype. Those are maybe the 6 most important, most basic words relating to genetics. So if you don't remember what any of those words mean, listen for that as we go through here and see if you can figure out what those words mean. Okay, then we'll come back and talk a little more about Mendel's work and some of the important things that he figured out about genetics. Hang on, I know what I need to do. We're going to try out my theory. All right, leave that one. That on too, so you can see your notes. Okay. I need to go on mute. Hang on. There we go. Okay, so that reviewed, like I said, a bunch of the really basic vocab. Hopefully that all sounds familiar to you. If any of that is vocab that you don't really remember, or you listen to that and you were like, oh yeah, I remember hearing that once, but I'm not going to remember all of it. This picture links to that video in the notes that are posted under the Unit 9 resources in Classroom. So you can go back and watch that and actually write down some definitions of phenotype and genotype and homozygous and heterozygous if you need to do that. Because those are words that are really important in genetics and things that we'll be talking about a bunch. Today we're going to move on and talk a little bit about what Mendel figured out. So he was a scientist who actually is pretty amazing, because when he was doing his work, we didn't know anything about DNA. We didn't know how information was passed on. We didn't know what DNA was, much less that it had these base sequences and how they coded for things. All he really had to work with were the physical characteristics that he could see, the phenotypes of those pea plants that he was working with. But from the patterns that he saw over and over and how traits were passed on, he was able to figure out quite a lot about how those traits are inherited. So this is kind of the model that he came up with to explain the patterns that he saw. First of all, he realized that there had to be something that was carrying information from parents to offspring. He called that a gene, and he realized that there were different variations of those genes. We call those different versions of genes alleles. And those account for variation in inherited characters. The word character is similar to the word trait. There is a very technical difference that you don't need to worry about so much, but when you hear the word character in this kind of sense, it essentially means a trait. So an inherited trait, some characteristic, that's basically where this word came from, that you can observe either externally. It is important to remember that genetic traits can be internal as well. It can be things that have to do with the chemistry of how your body works, how a certain protein is expressed in your body, how fast your metabolism is, how efficiently your blood transports oxygen. All of those things can have a genetic component too. So it doesn't have to be physically visible on the outside. A lot of the examples that we use are, just because they're the easiest to kind of visualize and think about. For each character or trait, each organism inherits 2 copies of a gene, one from each parent, at least when we are talking about sexually reproducing organisms. Because, of course, if you're talking about something like bacteria, they don't have 2 parents. They're haploid. They have 1 copy of each gene that they pass on. But we're going to start talking here mostly about inheritance within people. And so we're talking about sexually reproducing organisms. So you have 2 copies of every allele, one that you got from your mom and one that you got from your dad. If those 2 alleles are different, the dominant one determines the phenotype. Now, it is important to note here, we are talking right now about Mendel's gene model. And kind of just by chance, all of the characteristics that he looked at in his pea plants had one allele that was clearly dominant over the other. Not every gene works this way, and as we go through next week, we'll get into some that are a little more complicated than just dominant and recessive. But this was kind of the first understanding, is that one allele was dominant and one was recessive, and that is true for certain traits. He also came up with something called the law of segregation, which basically means out of those 2 copies that you have, one from mom, one from dad, only one of them gets passed on to the offspring. And so at some point, we now know that happens during the process of meiosis and the formation of egg and sperm cells or gametes of any type. Those 2 alleles separate from each other and only one of them ends up in the reproductive cell to be passed on to the next generation. And which of the 2 alleles gets passed on to the offspring is random. This is one of the places that variation can occur, is in which of the 2 copies of the gene you get from your parents. Any questions about the basic parts of Mendel's gene model? Okay. Oh, that's just a definition of alleles. Remember, when we're talking about a gene, we're talking about a specific nucleotide sequence, so a specific sequence of A's, T's, C's, and G's at a certain location or locus on a chromosome. And the combination of alleles, A's, T's, C's, G's, the sequence of bases that makes green pea pods is slightly different than the sequence that makes yellow pea pods, but they're both located in the same part of the same chromosome. Often when people work with genetics, one thing that you will frequently hear about when you hear about genetics experiments is something called a test cross. And this is something that I want you to think about and actually turn and talk to somebody about for a minute. If you have a purple flowered plant, and you know purple is dominant and white is recessive, you have a plant with purple flowers. You want to find out whether it is homozygous dominant or heterozygous, right? Because both of those would give it purple color. You do not have a DNA sequencer where you can actually go through and read the bases. How could you figure out the genotype of this plant? Turn and talk about that for just a minute. I just need to change how I'm sharing this real quick so that they can see what I'm writing. All right. So what would you do? Okay, so the best way to do this would be to do something called a test cross. A test cross means you take whatever individual you want to test and you breed or cross it with a homozygous recessive individual. That's what a test cross is. So in this case, you have this purple flowered plant that could be homozygous dominant, or it could be heterozygous. I need to make my capital P's different somehow. P is not a great letter. If you cross this plant with a white flowered plant, which you know has to be homozygous recessive because that's the only way to see white, then you know, here we go with Punnett squares, you ready? If the homozygous dominant, or if the purple plant was homozygous dominant, so these will be my capitals, and we cross it with a recessive plant, what color are all these offspring going to be? Purple. They are all going to get a dominant allele from this parent, so you will have all purple flowers. So if you do that cross and you get all purple flowers, it's not 100% conclusive, but it's pretty darn conclusive that that was probably a homozygous dominant plant. If that purple plant was heterozygous, oh, I did not spread those out very well. Just draw my line carefully there. And we do our test cross. Now what would you expect in the results?
We have one.
It'd actually be more than that, because this one's homozygous recessive. You're thinking of two heterozygotes. So this one, so you'd have two purple and two white. So you should have about 50 and 50. Does that make sense to everybody? And obviously in real life, you're not going to have exactly 50-50. If you get, 50 seeds from that plant, you might have like 28 that are purple, and what would that be? 22 that are white. But if you see any white in there at all, you know that this parent had to have a recessive gene. So a test cross means breeding it with a homozygous recessive individual, because you know that recessive trait is going to be hidden. So basically the characteristics of those offspring are being determined by the individual that you want to learn about. Does that make sense? Because you know the other one's all recessive. So all of the traits are being determined by the other parent. Any questions about that idea? Because this is something you'll see come back a few times throughout the next week. Okay. One of the other things that Mendel came up with is called the law of independent assortment. And this is something else that's important when you're thinking about the amount of variety or variation that we get in the genetics of sexually reproducing organisms. Before I actually define this for you, I'm going to talk you through a little bit how Mendel figured this out and kind of what his thought process was. So first, a couple of vocab words that you'll hear and that you should be familiar with. A monohybrid. So hybrid always means that they have two different traits they got from two different parents or different genes. So in other words, in genetics, it means they're heterozygous. Mono means one. So in this case, they're heterozygous for one trait. So a monohybrid cross would be two heterozygous individuals that are being crossed. A dihybrid is an individual that's heterozygous for two traits. Di means two. So their genotype might look like this, capital A, lowercase a, capital B, lowercase b. So a dihybrid cross is when we cross two heterozygous individuals. We're not quite going to do that here, but the question here, independent assortment, has to do with whether the genes themselves are linked and travel together. So here's kind of what you need to remember. Remember, these genes are located on chromosomes, okay? So this is a chromosome. If gene A is right here, and this allele happens to, or this chromosome happens to carry the dominant allele, and if gene B is right here and also happens to be dominant, you would expect that entire chromosome to move together, right? And so the offspring that got the capital A would also get the capital B. What Mendel was kind of wondering is, like, do certain traits always stick together like that, or does every single gene kind of separate on its own and get inherited separately? This is how he figured that out. So say we have two dihybrid individuals. They're heterozygous for both traits. If these genes are connected to each other. So, for example, if the two dominant alleles are always together and the two recessive alleles are always together, this is kind of what would happen. That would mean that our Punnett square is actually pretty simple, because if these two always stay together and these two always stay together, there aren't all that many combinations. This parent is either going to pass on the two dominants or the two recessives. You with me? And the same for this parent. If that's how they're always linked together, in which case, if this is true, now if we fill out our Punnett square, you would now see three individuals who show the dominant phenotype and one who shows the recessive phenotype for both traits. Does that make sense to everybody? So you would expect to see a 3-to-1 ratio of the dominant to the recessive characteristics. That would be the prediction. If, however, they assort independently, do you guys remember how to do this? If any combination of A's and B's is possible, now we suddenly have a lot more options. If they can mix and match in any possible way, we have actually 4 different combinations. And do you remember when we're looking at, so we'll start with the genotype of the first parent. When we're looking at this parent right here, do you remember how to find the 4 combinations for that parent? There's a trick that I think most bio teachers that we stole from math. You foil it. Yep. So the combinations, anytime, you can do this anytime you have genotypes for 2 traits. So they could pass on the first A and the first B. They could pass on the outer A and B outside. They could pass on the 2 inside, or the last A and the last B. Does that make sense to everybody? In this case, this is a dihybrid cross. So both parents have the same genotype, so we're going to put those same letters down the left side. If the other parent had a different genotype, you would foil their genotype as well. But in this case, they're the same. Then we can go through and actually fill this out. A couple of tips or reminders. It doesn't actually matter, like here we're using A's and B's. It doesn't actually matter which letter you write first, but you should be consistent. So here we started with A's before B's. That's how you should do it all the way through your whole problem. It'll just make your brain hurt less. If you should always write the 2 A's together and then the 2 B's together. And if an organism is heterozygous, you always write the capital before the lowercase for that letter. Okay, those are just kind of the rules of the road for how we write genotypes. So we can go through and fill this in. So now if we go through and start looking at what the phenotypes of these offspring would be. We're no longer going to have a 3-to-1 ratio because we have a lot more than just dominant or recessive. We are going to have 9 offspring that show both dominant traits. That'll be these 1, 2, 3, 4, 5, 6, 7, 8, 9. All of those have one capital A at least and at least one capital B. So there's going to be 9 that show both dominant traits. Okay. We are going to have 3 that are dominant for the A trait, but recessive for the B trait. Those 3 right there. So if we're writing this as a ratio, so far we're 9-to-3. And then you have 3 that are kind of the opposite. They're recessive for A, but dominant for B. And the last one, you have 1 out of 16 that's going to be recessive for both traits. So a dihybrid cross where both of the parents are heterozygous for both traits will always give you this 9-to-3 to 3-to-1 ratio in the phenotypes of the offspring. And so now we have kind of these 2 hypotheses, and just like with the test cross before, we could actually cross these 2 organisms, and Mendel did crosses like this to find out whether these 2 genes were linked. Remember, that was our original question, is do capital A and capital B always go together? And of course, when he did this, he found that this was not true. This did not happen. You did not get this 3-to-1 ratio. You got all the diversity that we see in this big, huge dihybrid cross. He saw that 9-to-3 to 3-to-1 ratio. And so he realized that every single gene acts on its own and separates independently from every other. In practical terms, this should kind of make sense if you think about some of the traits of your body and your appearance that are genetically determined. It doesn't make any difference whether you have blue eyes or brown eyes or gray eyes or green eyes. That has absolutely no effect on how tall you are, right? And whether or not you have a widow's peak, your hairline comes to a point that has absolutely no influence on whether or not you have a hitchhiker's thumb, right? It seems silly to even say those kinds of things, that it would matter, but if some of these genes were linked, were connected like that, then we would expect to see some of those traits go together all the time. So this type of research, coming up with these hypotheses and then actually doing this cross to see which one happened, led Mendel to come up with the law of independent assortment. Can I erase this? Feel okay? Oh, wait. So his law of independent assortment states that 2 or more genes assort independently. In other words, the alleles of one gene segregates and goes off to one egg or the other, or one sperm or the other, independently without any impact on other alleles during the formation of sperm and eggs. Now again, there are exceptions, as with most things, and as we've learned more about how genetics and biology works. This is true of genes located on different chromosomes. Obviously, now that we know what chromosomes are and where DNA is, it's easy to see that a gene on this chromosome is not going to affect one up here. It's also true of genes that are kind of on opposite ends of the same chromosome because of crossing over, which we'll talk about later. But there's so much crossing over that happens that if genes are on opposite ends of the same chromosome, they usually get separated. And so they also sort independently, which means any combination of genes is possible in the gametes that are being passed on to the offspring. Questions at all? All right, so that is the end of what we're doing for notes today. Most of today was actually review of stuff that you've hopefully heard before, maybe with a couple new words thrown in there. Spend some time reviewing over the weekend. If you feel like your genetics background is a little rusty, go look at the prior knowledge document for chapter 11 and review that. Also, throughout the next few days, I am going to be providing you with some basic practice problems, and they'll get more complicated as we go. But on today's classroom post, there's a PDF with some genetics practice problems, all of which are just basic Punnett square questions, except the very last one, which kind of starts looking at what we're going to do Monday. So consider that one kind of an extra challenge. There's also an answer key linked online. These are not assigned as homework, and they're not things I'm going to collect and grade, but you should be comfortable with doing Punnett square problems, especially these really basic ones. So if you want to pull that up and just do a few practice problems in your notebook and then go and check your answers, of course, come and let me know if there's anything that you have questions about, okay? So for the last 10 minutes today, you're welcome to pull those up and work on some of those problems right now. I don't know if 10 minutes is enough for those of you in class to start any test corrections. Probably, maybe not. I don't know. But that's all we got for today. Any questions? Okay, so Kylie and Ashley, if you have any questions, you can let me know. Otherwise, you are welcome to sign off. Go try some of those practice problems, and we can talk about on Monday if there's anything you had questions about, okay? Have a good weekend. Yes, I do.